So we are simultaneously working on a paper on schizophrenia right now that is basically using the single cell profiling technologies, using these editing and manipulation technologies to basically show how the master regulators underlying changes in the brain that are sort of found in schizophrenia are in fact affecting excitatory neurons and inhibitory neurons in pathways that are active both in synaptic pruning but also in early development. We've basically found a set of 4 regulators that are connecting these 2 processes that were previously separate in schizophrenia, in sort of having a sort of more unified view across those two sides. The second one is in the area of metabolism. We basically now have a beautiful collaboration with the Goodyear lab that's basically looking at multi-tissue perturbations in 6 or 7 different tissues across the body in the context of exercise and in the context of nutritional interventions using both mouse and human, where we can basically see what are the cell-to-cell communications that are changing across them. And what we're finding is this immense role of both immune cells as well as adipocyte stem cells in sort of reshaping that circuitry of all of these different tissues. And that's sort of painting to a new path for therapeutical interventions there. In Alzheimer's, it's this huge focus on microglia. And now we're discovering different classes of microglial cells that are basically either synaptic or immune. And these are playing vastly different roles in Alzheimer's versus in schizophrenia. And what we're finding is this immense complexity as you go further and further down of how, in fact, there's 10 different types of microglia, each with their own sort of expression programs. We used to think of them as, oh, yeah, they're microglia. But in fact, now we're realizing just even in that sort of least abundant of cell types, there's this incredible diversity there. The differences between brain regions is another sort of major insight. Again, one would think that, astrocytes are astrocytes no matter where they are. But no, there's incredible region-specific differences in the expression patterns of all of the major brain cell types across different brain regions. So basically, there's the neocortical regions that are sort of the recent innovation that makes us so different from all other species. There's the sort of, reptilian brain sort of regions that are sort of much more very extremely distinct. There's the cerebellum. There's each of those basically is associated in a different way with disease. And what we're doing now is looking into pseudotemporal models for how disease progresses across different regions of the brain. Mm-hmm. If you look at Alzheimer's, it basically starts in this small region called the entorhinal cortex, and then it spreads through the brain and, through the hippocampus and, ultimately affecting the neocortex. And with every brain region that it hits, it basically has a different impact on the cognitive and, memory aspects, orientation, short-term memory, long-term memory, etc., which is, dramatically affecting the cognitive path that the individuals go through. So what we're doing now is creating these computational models for ordering the cells and the regions and the individuals according to their ability to predict Alzheimer's disease. So we can have a cell-level predictor of pathology that allows us to now create a temporal time course that tells us when every gene turns on along this pathology progression, and then trace that across regions and pathological measures that are region-specific, but also cognitive measures and so on and so forth. So that allows us to now sort of, for the first time, look at, can we actually do early intervention for Alzheimer's, where we know that the disease starts manifesting for 10 years before you actually have your first cognitive loss. Can we start seeing that path to build new diagnostics, new prognostics, new biomarkers for this sort of early intervention in Alzheimer's? The other aspect that we're looking at is mosaicism. We talked about the common variants and the rare variants, but in addition to those rare variants, as your initial cell that forms the zygote divides and divides and divides, with every cell division, there are additional mutations that are happening. So what you end up with is your brain being a mosaic of multiple different types of genetic underpinnings. Some cells contain a mutation that other cells don't have. So every human has the common variants that all of us carry to some degree. the rare variants that your immediate tree of the human species carries. And then there's the somatic variant, which is the tree that happened after the zygote that sort of forms your own body. So these somatic alterations is something that has been previously inaccessible to study in human postmortem samples. But right now, with the advent of single-cell RNA sequencing, in this particular case, we're using the well-based sequencing, which is much more expensive, but gives you a lot richer information about each of those transcripts. So we're using now that richer information to infer mutations that have happened in each of the thousands of genes that sort of are active in these cells, and then understand how the genome relates to the function, this genotype-phenotype relationship that we usually build in GWAS in genome-wide association studies between genetic variation and disease. We're now building that at the cell level, where for every cell, we can relate the unique specific genome of that cell with the expression patterns of that cell and the predicted function using these predictive models that I mentioned before on dysregulation for cognition, for pathology in Alzheimer's at the cell level. And what we're finding is that the genes that are altered and the genetic regions that are altered in common variants versus rare variants versus somatic variants are actually very different from each other. The somatic variants are pointing to neuronal energetics and oligodendrocyte functions that are not visible in the genetic lesions that you find for the common variants, probably because they have too strong of an effect that evolution is just not tolerating them on the common side of the allele frequency spectrum.
So the somatic one, that's the variation that happens after the zygote, after
Correct.
For you individual. I mean, it's a dumb question, but there's mutation and variation, I guess, that happens there. And you're saying that through this, if we focus in on individual cells, we're able to detect a story that's interesting there, and that might be a very unique kind of important variability that arises for, you said neuronal or something that would sound
energetics.
Energetics. It sounds like a cool term.
So I mean, the metabolism of humans is dramatically altered from that of nearby species. We talked about that last time, that basically we are able to consume meat that is incredibly energy rich, and that allows us to sort of have functions that are, meeting this humongous brain that we have. So basically, on one hand, every one of our brain cells is much more energy efficient than our neighbors, than our relatives. Number two, we have way more of these cells. And number three, we have, this new diet that allows us to now feed all these needs. That basically creates a massive amount of damage, oxidative damage, from this huge, super-powered factory of ideas and thoughts that we carry in our skull. And that factory has energetic needs, and there's a lot of sort of biological processes underlying that we are finding are altered in the context of Alzheimer's disease.
That's fascinating. So you have to consider all of these systems if you want to understand even something like diseases that you would maybe traditionally associate with just the particular cells of the brain.
Yeah.
The immune system,
the metabolic
system.
And these are all the things that makes us uniquely human. So our immune system is dramatically different from that of our neighbors. Our societies are so much more clustered. The history of infections that have plagued the human population is, dramatically different from every other species. The way that our society and our population has sort of exploded has basically put unique pressures on our immune system. And our immune system has both coped with that density and also been shaped by, as I mentioned, the vast amount of death that has happened in the Black Plague and other sort of selective events in human history, famines, ice ages, and so forth. So that's number one, on the sort of immune side. On the metabolic side, again, we are able to sort of run marathons. I don't know if you remember the sort of human versus horse experiment, where the horse actually tires out faster than the human, and the human actually wins. So on the metabolic side, we're dramatically different. On the immune side, we're dramatically different. On the brain side, again, no need to sort of, it's a no-brainer how our brain is like just enormously more capable. And then in the side of cancer, so basically the cancers that humans are having, the exposures, the environmental exposures is, again, dramatically different. And the lifespan, the expansion of human lifespan is unseen in any other species in, recent evolutionary history. And that now leads to a lot of new disorders that are starting to, manifest late in life. So, Alzheimer's is one example where basically, these vast energetic needs over a lifetime of thinking can basically lead to all of these debris and eventually saturate the system and lead to, Alzheimer's in the late life. But there's just such a dramatic set of frontiers when it comes to aging research that will. So what I often like to say is that if you want to engineer a car to go from 70 miles an hour to 120 miles an hour, that's fine. You can basically fix a few components. If you want it to now go at 400 miles an hour, you have to completely redesign the entire car because the system is just not evolved to go that far. Basically, our human body has only evolved to live to, 120. Maybe we can get to 150 with minor changes. But if, as we start pushing these frontiers for not just living, but well living, the F-zine that we talked about last time. So to basically push F-zine into the 80s and 90s and 100s and, much further than that, we will face new challenges that have, never been faced before. In terms of cancer, the number of divisions, in terms of Alzheimer's and brain-related disorders, in terms of metabolic disorders, in terms of regeneration. There's just so many different frontiers ahead of us. So I am thrilled about where we're heading. So basically, I see this confluence in my lab and many other labs of AI, of, the next frontier of AI for drug design. So basically these sort of graph neural networks on specific chemical designs that allow you to create new generations of therapeutics. These molecular biology tricks for intervening at the system at every level. This personalized medicine, prediction, diagnosis, and prognosis using the electronic health records and using these polygenic risk scores, weighted by the burden, the number of mutations that are accumulating across common, rare, and somatic variants, the burden converging across all of these different molecular pathways, the delivery of specific drugs and specific interventions into specific cell types. And again, you've talked with Bob Langer about this. There's, many giants in that field. And then the last concept is not intervening at the single gene level. I want you to sort of conceptualize the concept of an on-target side effect. What is an on-target side effect? An off-target side effect is when you design a molecule to target one gene and instead it targets another gene and you have side effects because of that. An on-target side effect is when your molecule does exactly what you were expecting, but that gene is pleiotropic. Pleio means many, tropos means ways, many ways. It acts in many ways. So it's a multifunctional gene. So you find that this gene plays a role in this, but as we talked about, the wiring of genes to phenotypes is extremely dense and extremely complex. So the next stage of intervention will be intervening not at the gene level, but at the network level. Intervening at the set of pathways and the set of genes with multi-input perturbations to the system, multi-input modulations, pharmaceutical or other interventional, that basically allow you to now work at the sort of full level of understanding, not just in your brain, but across your body, not just in one gene, but across the set of pathways, and so on and so forth for every one of these disorders. So I think that we're finally at the level of systems medicine, of basically instead of sort of medicine being at the single gene level, medicine being at the systems level, where it can be personalized based on the specific set of genetic markers and genetic perturbations that you are either born with or that you have developed during your lifetime, your unique set of exposures, your unique set of biomarkers, and your unique set of current set of conditions through your EHR and other ways. And the precision component of intervening extremely precisely in the specific pathways and the specific combinations of genes that should be modulated to sort of bring you from the disease state to the physiologically normal state, or even to a physiologically improved state through this combination of interventions. So that's in my view the field where basically computer science comes together with artificial intelligence, statistics, all of these other tools, molecular biology technologies and biotechnology and pharmaceutical technologies that are sort of revolutionizing the way of intervention. And of course, this massive amount of molecular biology and data gathering and generation and perturbation in massively parallel ways. So there's no better way, there's no better, time, there's no better place to be sort of, looking at this whole confluence of ideas. And I'm just so thrilled to be a small part of this amazing, enormous ecosystem.
Yeah, it's exciting to imagine what humans of 100, 200 years from now, what their life experience is like, because these ideas seem to have potential to transform the quality of life. That when they look back at us, they probably wonder how we were put up with all this suffering in the world.